And thank you for the organizers for inviting me. So today I will share with you some of the work we've been doing to try to understand how we can translate a new class of antibodies for cancer therapy, namely the IgE class, and how we have found, what information we have found that influence the antibodies of these types of this class can influence the tumor microenvironment. What interests a lot our group is the ability of antibodies to engage through the Fc region with effector cells, such as macrophages and monocytes and NK cells, to trigger effector functions such as antibody-dependent cell-mediated cytotoxicity and phagocytosis. And we also, a stream of work in our lab is also looking at conjugating antibodies to toxins to develop antibody drug conjugates. But today I will focus more on the Fc portion of the antibody and how we can influence the tumor microenvironment by changing this Fc region. And we have focused a little bit more in our lab on ovarian cancer and also on melanoma, and I will share some of these insights with you. For ovarian cancer, one of the huge motivations for us is that normally this disease is diagnosed at a very late stage 3 or 4, at which point the 5-year survival, as you can see, is very, very poor. Whereas if this cancer can be detected earlier, one can see that the survival is better. However, now we have a challenge where the tumor microenvironment is modulated and not immunoactive. And we want to change the tables on cancer by changing the way we engineer antibodies to influence the microenvironment. So one classic train of thought in our lab is that looking at the antibodies that are now available for cancer therapy, we can clearly see that almost all of them are of the IgG class. And in fact, IgG1 is the most prominent antibody class used. One wants effector functions and Fc-mediated the immune microenvironment. And many tumors of the hematopoietic nature are benefiting from IgG antibodies, and also solid tumors are starting to emerge now more and more, especially with the checkpoint inhibitor immunotherapy era that we are in. But interestingly, looking at the antibody known mechanisms of action, one of the key mechanisms of actions is through the Fc region, cytotoxicity, phagocytosis, complement activation, etc. And that even includes a checkpoint inhibitor such as anti-PD-L1 and anti-CTLA4 for many reasons. So we figured that it is possible that we can look at how we can enhance and activate those effector functions in the tumor microenvironment to activate important sentinels. However, we do know from our own work that the tumor microenvironment has Fc receptors that are inhibitory for IgG class antibodies, which are already the majority of antibodies that are found that are available in the clinic for cancer patients. And we know that the tumor microenvironment also has B cells that express alternatively activated classes of antibodies such as IgG4, and these are negative regulators of immunity, and they can also interfere with otherwise such toxic antibodies such as IgG1 that are used for cancer therapy, for which we expect to have effector functions. And interestingly, important immune sentinels in the tumor microenvironment, such as macrophages, which sometimes comprise more than 30% of immune infiltrate in tumors, are actually alternatively activated, expressing scavenger receptors such as CD163. But what we want to do is to engineer antibodies that are not prone to immune blockade and that can reactivate the tumor microenvironment, including cells like macrophages. So therefore being able to target tissue cancer biology to turn the tables against cancer. And so what we wanted to do is we're understanding that IgG antibodies work very effectively in the circulation. They have a very long half-life of 2 to 3 weeks in the circulation, but they're less immunoactive in tissues. And can we then engineer an antibody that is naturally immunoactive in tissues? It's able to operate in Th2 bias conditions where IgG is less able to be activated because of inhibitor receptor, Fc receptor expression, and not compete with other IgGs for Fc receptor binding and activation. And could it activate immune sentinels in the tumor microenvironment, such as monocytes and macrophages, so influencing essentially this interaction between the Fc region and the immune cells, such as macrophages. And one clear candidate in our mind is an antibody class that actually works and is immunoactive in tissues and works in Th2 environments such as IgE. And could this class activate macrophages that sit in the periphery of tumors such as ovarian cancers and melanomas and others? So all of us would, when we think of IgE, we think of allergy, we think of anaphylaxis, we think of Th2 immunity. And indeed, IgE antibodies are known for being having a pathogenic role in allergy. However, naturally, IgE antibodies are expressed in tissues in our bodies and help to activate immunity to protect from large pathogens such as parasites. And indeed, they're well known to have extremely high affinity for their own cognate receptors, Fc epsilon receptors, Fc epsilon R1 and Fc epsilon R2, also called CD23. This engagement has the affinity of 100 to 10,000 times higher for its receptors of IgE compared to IgG for its own Fc gamma receptors. That means that IgE is the only antibody class that sits on the surface of immune cells and surveys the body in the absence of antigen target all the time. This high affinity means it persists. So therefore, if you can target a tumor antigen with an IgE, you will have prolonged immune surveillance. Also, IgE antibodies are known to be potent inflammatory mediators of immune responses, triggering inflammatory cytokine release, recruiting of immune cells in tissues, and they're known to trigger classic effector functions such as ADCC and ADCP cytotoxicity phagocytosis against parasites. And we know that they're very potent triggers of antigen presentation, and the resulting immune response is not just an IgE, but also an across the board IgG spectrum response as well. And we know that IgE receptor expressing effector cells can also infiltrate tumors. Interestingly, unlike IgG, there is no Fc gamma R2b inhibitor receptor equivalent for IgE, and therefore the button is pressed on. And we wanted to understand how that could be applied in the context of cancer. So we set out to engineer IgE antibodies against cancer antigens in our hands. So we selected an antigen for many reasons. I cannot describe all of the reasons today, but it is this target is called folate receptor alpha. It is a folate carrier, and it's expressed at high levels, very highly overexpressed on the surface of certain types of tumors, such as ovarian cancer, mesothelioma, breast cancer, especially the basal type breast cancers and others. This target is present at all stages of a disease in different tumor types, including ovarian cancer, and is a marker for poor prognosis for patients generally. It is rarely expressed in normal tissues, and in fact, it's expressed in the apical surfaces of certain tissues, such as the kidney and the choroid plexus in the brain, which means that the access to from the blood circulation is much lower for those expressing tissues. And indeed, it's an emerging target with numerous therapeutic agents targeted against folate receptor alpha, including IgG and IgG1 antibodies with human Fc regions. So we selected one antibody, an IgG1 isotype targeting folate receptor alpha, and we re-engineered it with the Fc regions of the human IgE class. And we set out to study these molecules in the context of cancer therapy using different models. Our models were very difficult to select because IgE immunology is very different between humans and mice. And we knew that in a mouse model, human IgE Fc would not cross-react effectively at all with Fc epsilon receptors in mice. And indeed, the Fc epsilon receptor distribution in mice, it is not the same as in humans. And therefore, using a mouse equivalent syngeneic system wouldn't be of benefit in our studies. So what we did is we made a xenograft model of a human ovarian carcinoma in mice, where we engineered the human IgE and IgG equivalent antibodies, and we introduced human immune effector cells, which partly immunohumanize these mice, and they arrive in the tumors. So what we did is we then added the antibody together with effector cells with only one injection, and we try to see whether we can see follow tumor growth and measure tumor growth over time. Indeed, IgG had an effect, as you can see, compared to controls in the beginning with PBMCs with human immune cells. And we could see that was the case in the beginning at 19-20 days, but then on long term, the tumor started to grow almost the same size as the controls. Even at half that dose, IgE was able to restrict tumor growth at early on, but also later on in the course of the disease. But indeed, with full dose of IgE equivalent to the IgG, we could see that tumor growth was impaired. So we thought that's an interesting model. We are very happy with the results, but then we wanted to understand if that makes any difference in the context of patient cancer. So here we selected ovarian carcinoma patient-derived model, a PDX, that came from a patient with ovarian cancer at stage 3 and grade 3 tumor. And here again, we introduced the antibody together with effector cells, and these tumors grow in the peritoneal cavity of the mice like as ascites, and the animals have a turnaround time of about 20-24 days. And here, 2 injections, so 2 weeks apart, showed us that IgE was able to double the survival of the mice that received this treatment in these 2 doses. And in this particular model, IgG was not effective. Then we wanted to develop a model that has a more syngeneic immunocompetent setting to mimic more the patient situation, and we selected the rat for this. And the reason why we selected the rat was because the rat Fc epsilon receptor distribution is pretty similar to humans, expressed on monocytes, macrophages, eosinophils, dendritic cells, and many, many others. Mast cells and basophils, of course. So here we re-engineered our antibodies to have Fc regions of the rat species. So, and then we used the tumor that was designed from the same species. So it was in a syngeneic setting. So the tumors grow, as you can see here in the white dots, as lung metastasis in this model. And you can do this by staining with India ink anti-staining, which makes the white dots appear on the lungs, and one can measure the occupancy of tumor in the lung and also the number of lung metastases. And in this model, we did not have to reconstitute with immune cells because immune cells were found naturally in different parts of the body, as they would do in an immunocompetent host, and the tumor grew in the lung. So here we introduced weekly doses of the antibodies of IgG and IgE of the rat species, and we can see that IgG and IgE were able to restrict tumor growth, both in terms of the lung metastasis per square area of the lung, but also the percent of tumor occupying the lung. And we can clearly see that IgE was more effective than IgG, whereas IgG was also more effective than control. And you can see the pictures here, the lung metastasis distribution being lower compared to control for IgG, and IgE had very low occupancy compared to IgG and compared to control. This gave us 3 models where we could clearly see that in the new macroenvironment, or that the tumors were restricted in their growth when IgE antibodies were introduced. And indeed, I will show you some data later where the tumor cells in green are actually pulled apart by monocytes when we studied the effects of the antibody in vitro as well. What we found interesting is looking at the tumors from the patient-derived PDX model, we could see that the remaining tumors in the IgG-treated group appear to have an infiltration of human macrophages around the tumor area. And that was specifically only for the IgE-treated animals, but not the controls. So we suspected that monocytes, macrophages may be important effector cells in IgE anti-tumor response. And this was something that intrigued us a lot because we knew that monocytes and macrophages are found in tumor microenvironments in ovarian cancer and other solid tumors. So we set out to ask the question whether monocytes and macrophages might have an interesting contribution to anti-tumor functions of IgE. And in this model, we knew that if we mixed our human immune cells together with IgE in the PDX model, we see that the mice survive twice as long as the controls. So what we did is we depleted our PBMCs of monocytes. So all the other cells were introduced, and we could see that IgE at that point had very little effect in restricting tumor growth, and animals died the same amount of time or reached the levels that we considered lethal at 21 days, very close to those of controls. What we then did is, in the depleted PBMCs, we reconstituted them with the same concentration of monocytes, and we introduced those back into the animals together with IgE, and we could see that we could recapitulate this survival advantage that IgE had. This pointed to us that monocytes and macrophages may be important effector cells being triggered by IgE in the context of anti-tumor immunity. And as I mentioned before, we took that in vitro to show that in red here we have calcine AM stained tumor cells, and red we stained monocytes in a PBMC preparation ex vivo, and we put the antibody together with the cancer cells and the immune cells. Within 30 minutes, we could see the contact between tumor cells in green and monocytes in red out of all the other immune populations that we introduced in the culture dish. And within 90 minutes, we see this pulling apart of the tumor cells and contact with immune monocytes. And we could see the monocytes are starting to phagocytose bits of the tumor cells. So you can see the tumor cells are now falling apart and being pulled apart. Within 3 hours, we see remnants of tumor cells remaining and monocytes having ingested bits of them, of the tumor cells inside them are still in contact with those dying or dead cancer cells. In the control antibody, where we introduced the hapten-specific IgE, we see occasional contact between the immune cells, monocytes, and tumor cells, but here we see that the tumor cells appear to be green and still highly viable. Then we wanted to know whether it's possible that we can show killing of tumor cells ex vivo using patient-derived immune cells such as PBMCs and monocytes. And indeed, we selected PBMCs from patients with ovarian cancer who had folate receptor alpha positive and negative disease. And we could see that immune cells from any patient group in our cohort could kill tumor cells ex vivo in the context of anti-tumor IgE to folate receptor alpha. And you can see that here compared to controls, IgE, you can see killing by cytotoxicity. We wanted to know whether having one antibody that was against the folate receptor alpha target had this effect, but we didn't know whether that effect could be true for other antibodies against different targets. And here we decided to design a second-in-class IgE antibody against a different target. And this was against the target called chondroid sulfate proteoglycan 4, which is found overexpressed in cutaneous melanoma, gliomas, and other tumors. And indeed, we did a screen of immunohistochemistry to show that CSPG4 is expressed on the surface of tumor cells in a variety of melanomas, and it is not expressed on normal skin. And we can see that it's expressed in melanomas, on the skin, in lymph node metastasis, but also in distal metastasis, visceral metastasis in the body, and very little expression was found in the skin. So we engineered a humanized version of a mouse antibody to CSPG4 in an IgE format, and we could purify and produce it at decent quantities to be able to test. So then we did the basic experiment, as I explained before, where we used patient-derived immune cells, and we asked whether these immune cells can trigger ADCC against melanoma cells expressing the target. And clearly we can see very nice killing by patient-derived immune cells against two target cell lines that expressed the target CSPG4. We also use the classic mast cell degranulation assay to show that our antibody, together with target immune cells, target cancer cells on the other end, could trigger degranulation of mast cells. And that was really true as compared to control IgE. Cancer-specific IgE triggered degranulation of mast cells. So this antibody was functional through the Fc region and triggered effector functions. Indeed, we asked the same question as we did, the same question as we did before. We designed the melanoma subcutaneous model reconstituted with human immune cells, and we gave doses of an IgG equivalent and Ig equivalent to CSPG4, and we could see that if we gave a weekly dose, both IgG had an effect compared to controls, but also the IgE had an effect which was significantly better than the IgG at weekly doses. When we gave the antibodies every 2 weeks, only IgE had an effect, but IgG did not have a significantly different effect in restricting tumor growth in this model. Interestingly, as we mentioned before, macrophages were appearing in the tumor lesions that remained after treatments in the IgE cohort and also in the IgG cohort. We also asked whether it's possible that it's in this similar model, whether a lung metastasis model can also be targeted with IgE, and we showed that our tumor-specific IgE restricted the amount of tumors on the lungs of the animals compared to controls. And we were able to show that if we use human patient-derived immune cells from patients with melanoma, we could see that the tumors were restricted when we used our cancer-specific IgE antibody. And here in this last model, we used the patient-derived patient-derived xenograft model designed from 2 patients' tumors with melanoma, using autologous immune cells from the same patient in this model. And we could see that this the introduction of IgE restricted improved the survival of these mice over time compared to controls. So this way we were able to show that 2 different antibodies of the IgE class against 2 separate targets in 2 different indications had an effect against cancer growth. But we were very intrigued about this macrophage infiltration and these mechanisms, and we wanted to understand this a little bit more. So going back to our original anti-folate receptor alpha antibody, I will remind you that we were using PBS, IgG and IgE in immunocompetency and genetics system in the rat, and we could see that there is restriction of lung metastasis. We could show that looking at the tumor lesions and slicing through and staining for the target antigen, folate receptor alpha in green, but also the CD68 marker, which is a marker for macrophages. In this rat model, we can see that macrophages always surround the tumor lesions in the lungs of these animals, and these lesions express the target antigen folate receptor alpha. When we introduced IgG antibodies, we could see that this macrophage infiltration, as we have seen in the other models, appeared to be very well described. You can see here red cells, macrophages surrounding the tumor. What we found in the IgE treated animals was very different. We found that these tumor lesions were indeed swimming in macrophage infiltration. You can see here in red, and the tumor lesion are these foci in the middle here. These basically remnants or remaining tumor islands still surviving in the lesion were circled by this very big immune infiltrate. And indeed, the pathologists who looked at these lesions using hematoxin and eosine staining could see this effect as well. This is a control animal. You can see the tumor lesions in dark purple, very, very closely packed, and there's an immune infiltrate around. But if you look at the IgE treated animals, you can see the tumor lesions are swimming in this immune infiltrate around in the smaller tumors. So we took some of these tumors and we did a transcriptomic analysis to try to understand what's special about what's going on, the immune and the pathways that are activated or not activated in these animals with treatment with IgE. And to our surprise, we found that the control animals had upregulated pathways to do with cell cycle activation and regulation and proliferation, whereas the IgE treated animals appear to have immune pathway activation that was very prominent. And some of these pathways intrigued us because IgE antibodies, as everybody knows, have a role in allergy, and we were considering whether an allergic inflammation or activation could be triggered. But indeed, the pathways we see here activated are NK cells, IL-7, IL-12, which T-cell receptor, CXCR4, which is a recruitment for immune cells and triggering of tertiary lymphoid structure formation. So indeed, we found, as you can see here, IL-12 and NK cell activation. These were not classic mediators of allergy. These were more activators of classical pro-inflammatory immunity. So we were very intrigued by this, and we then wanted to know whether macrophages we can take outside the body and we can derive them. Are they also able to trigger tumor growth? And indeed, we could see that quiescent macrophages, M1 macrophages, and M2 macrophages, which are more likely to find in the tumor microenvironment, could all be triggered to kill tumor cells ex vivo by IgE. And this was a surprise to us because we considered that M2 alternatively activated macrophages, which are found in tumors, are less activatory. And maybe what we're seeing here is a repolarization and activation of macrophages against tumors with an IgE class antibodies. And indeed, when we looked at the cytokine milia that was found in the tumor microenvironment and in the context of anti-tumor immunity triggered by IgE, we found that these macrophages, especially the M2 phenotype, but also the M1, had a very interesting pro-inflammatory mediator activation, TNF-alpha, interferon gamma, IL-12, IL-1 beta, not IL-4, but also IL-10, MCP1 was a massive chemoattractant for macrophages, and RANTES. These are mediators of a unique phenotype, which doesn't fit within the M1 and M2 categories, but fits into what we consider an alternatively a newly polarized macrophage phenotype. And we consider that a tumor microenvironment has repolarize in the context of IgE, especially the alternatively activated phenotypes. So what I would like to summarize for you is that we found that anti-tumor IgE has demonstrated an in vivo superiority over the IgG counterparts in our models, and in different models indeed, and in different indications. We found in vitro and ex vivo studies that IgE can direct monocyte recruitment against tumors and macrophage activation, and there's an interesting mediator axis polarizing macrophages towards a pro-inflammatory TNF-alpha rich and MCP1 triggered axis, which recruits macrophages into the tumor lesion. And this is something we've studied a lot in the lab. I cannot describe all of it today, but I will give you the flavor that we found, that M2 macrophages become this newly polarized state, whereas M1 macrophages appear to retain their activatory phenotype in the context of IgE, and they can trigger effector functions in the context of IgE. Some of this work has now resulted in a clinical trial for the first-in-class IgE antibody against cancer, which is undergoing a clinical trial at Guy's Hospital, with other sites as well coming on board. And our work and our insights on IgE immunology against cancer has contributed to a field called allergo-oncology, which we are personally contributing to, and it's supported by the European Academy of Allergy and Clinical Immunology, in which we are a task force and we have activities looking at the tumor microenvironment, epidemiology, medicine, oncology, and many other disciplines in this context. And myself, I was able to launch a company called IGEM, which is now called Epsilogen, which focuses on immuno-oncology using IgE therapeutics for cancer therapy, taking the concept further away, not just in the clinical trial, but perhaps into a larger cohort of patients who might benefit. Our lab is very much interested in B-cell immunology, the antibody responses in cancer, and we have a range of skills in the lab, ranging from basic immunology, IgE engineer, antibody engineering of different isotypes, and translating our work through development and safety and into clinical studies and now commercialization. And we do this using a variety of tools and collaborating with many different clinicians from different disciplines such as dermatology, gynecologic oncology, clinical trials, allergy, and others. And also collaborating with colleagues at Cancer Research UK who are funding some of this work, but also with labs such as colleagues at the Royal Marsden, Queen Mary's University of London, and Medical University of Vienna, and others. And I'd like to thank all my colleagues and my lab for working with us on these topics. So I'd like to stop there, and I'm happy to take any questions.
Thank you, Professor Sophia, for a nice presentation that enlightened us the superiority in terms of results of using IgE over IgG in activating tumor microenvironment. And it's very interesting because we know that most of the challenges we are facing in treating cancer is the immunosuppressive tumor microenvironment. And the promising results which have been shown by optimization of IgE to activate the tumor microenvironment, I think is something which is very interesting. So I thank you so much for your nice presentation. And there is one question which have been posted by one of the participants, who she's asking, like, is there a risk of the immunotherapy becoming resistant over time in cancer patients when we are using immunotherapy? I think she's trying to compare with other traditional treatment. Please, you're welcome.
So the question is, so how do we overcome suppression of the tumor by the tumor microenvironment? Is that the question?
The question is, like, she's asking the risk of the targeted therapy or immunotherapy to develop a resistance when used over time.
Yeah, so as you can see, in this context, we have been giving very few injections in the animals. So the animals receive 1, 2, or 3, or 4 doses of the drug, and we see an effect. What we haven't done is we've given the drug consistently over many months, so we can see whether at some point the tumors develop resistance. I suspect that in like any therapies, a monotherapy will have at some point a stop button where there will be a resistance or at least a lack of response to any immunotherapy. So if the tumor microenvironment may be expanded or the tumors have been killed by a particular targeted therapy, then there will be a rise of tumors that are resistant to the therapy. A simple, very basic concept would be that folate receptor alpha, for example, therapy, the antibody, could kill all folate receptor alpha positive tumors, and then that allows for the rise of non-expressing folate receptor alpha expressing tumor cells, in which case a combination therapy or a follow-on therapy will have to be considered. Now, in terms of a tumor microenvironment, again, we don't know the long-term effects of triggering or repolarizing M2 macrophages into a newly polarized phenotype, and that's something we probably need to follow up and look at in different models, and also in patients with cancer who are now receiving the drug.
Dr. Sophia, I continuing the same question, this was asked by myself, Ramya. I was just taking up with the bevacizumab example, as you said, giving 1 or 2 doses to mice. What happens if the person receives more than an year of bevacizumab? And if person develops a resistance to it, as you said, the next cycle line will be like combining the 2 other drug again, or how does the tumor regress, or it will come back? How does it act?
I would not be able to answer this question. We've never combined anti-VEGF therapy with our antibodies. So I wouldn't know whether that is a viable therapy at all for IgE as a combination. And I also would not know, but I suspect that there may be a possibility to combine with checkpoint inhibitors, because we have seen some activation of checkpoint molecules in response to IgE and even IgG immunotherapy against cancer. So we consider that perhaps a combination with checkpoint inhibition may be a viable route. In terms of the anti-VEGF, I must say that we never really looked properly at the vasculature. Having said that, CSPG4 targets cancer cells on the surface, high expressing CSPG4 cells, but also CSPG4 is overexpressed in tumor-associated neovasculature, and it is possible because it's a stem-like, it's a neurocrest antigen, that you will have a very good clearance of blood vessel formation in response to a therapy like anti-CSPG4. But that remains to be seen and to be studied in the right models.
Okay, thank you.
Thank you.
Okay, can I have a question, please?
Sure, you're welcome.
Okay, Dr. Sophia, really for that nice presentation. My kind of question is like very simple. I just wanted to know, like there are certain diseases in which you've got like IgE excess, like allergic bronchitis and allergic responses. So once like those natural processes or natural course of disease allows some individual to have higher degree of IgE in their body, do you think does it protect them in any way in terms of this tumorigenesis or these carcinogenic processes, or some kind of protection they offer?
That is a really key question, and the million-dollar question in some ways. There's been a lot of epidemiological studies around high levels of IgE and consideration whether they are protective against the rise of cancer, or even levels of IgE within the patients who already have cancer. There have been some studies to show that there is a correlation between levels of IgE and protection from cancer or cancer growth in the future, and some studies show no. And the question sometimes is that what type of tumor are we talking about? It appears that certain types of tumors having high IgE ahead of time, over time, protects from the growth of certain types of tumors, but not others. In fact, there's some tumors, for example, lung cancer, where IgE high levels are considered not a good idea. But other types of cancer, like glioma, for instance, or some gynecological and skin cancers, where high IgE levels may be protective. So the jury is still out, but I do agree that having IgE levels at a normal level rather than abnormally high or whatever is a good thing. And there's been some studies from Albert Einstein in the past 4 or 5 years looking at these patients with ultra low IgE levels who are immunodeficient. And these patients, including children with these low IgE levels, appear to have a higher chance of getting certain types of tumors. So I think this is quite key, because I think IgE is part of a natural IgE immunological physiology and homeostasis. And if those levels drop, it might not be that you need very high levels, but it may be that you have normal levels. It's part of an immune surveillance to protect us from anything that goes wrong, including cells that are becoming cancerous. So I think the other way around is the thinking, whether having normal, physiologically normal levels of IgE is what we really need, and having very low levels or immunodeficient levels of IgE, we are not protected. So I think these are things, it's not my personal work, but I think it's very, very exciting work by other people. I'm following their research very closely.
Ahmed, thank you very much. I think there are like populations who have got like higher level of IgG in certain part of the world because of the so many parasitic and other infections.
Yes.
So I also feel that they've got less number of these tumors as well.
I don't think anybody has studied properly these patients, because they are different to those who have IgEs out of allergy, you see. And I don't think there's been enough studies. So I think somebody out there needs to be doing these studies, not us, it's not our area. We are more antibody engineers and immunologists. But I think studying these populations of people who have high IgE levels because of, parasite infection exposure, there may be a different population to study with a different outcome in relation to cancer. You see what I mean? Because this is a different type of immunity. IgE in allergy is a classic Th2 immunity, but IgE against parasites is an antiparasitic immunity, which is not a classic Th2.
Okay, fine. We need more, I think, specific studies in this regard. Thank you very much, Sophia. It was really a very nice presentation. Thank you very much.
Thank you.